Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Advancing LNP Characterization for Drug Delivery with NeoVac. This is session 3 of our virtual event, Biopharmaceutical Analytics Unveiled, Expert Insights and Customer Success Stories. I'm Roisin McGuigan, an editor at BioInsights, and joining me today I have Blaj Bacalar, Tristan Kovacic, and Dan Peer, who will discuss simplifying LNP characterization via simultaneous analysis of key attributes. After the presentations, we'll have a live Q&A session with Tristan and Blaj. So feel free to pose your questions using the Ask a Question box to the left of your screen, and we'll try to get to them during the session. Now, I'd like to introduce our presenters. Blaj is the PATfix product manager at Sartorius BIA Separations. Tristan is a scientist at Sartorius BIA Separations and specialises in process development and analytics for non-viral drug delivery systems and active pharmaceutical ingredients. Dan is chairman of NeoVac and director of the Laboratory of Precision Nanomedicine at Tel Aviv University. So now, without further ado, I'll hand over to Blaj to kick off today's presentations.
Thank you, Roisin, and welcome back, everybody. So we've done pDNA, we've done mRNA. Now let's go into final phases of the mRNA drug substance where it becomes a drug product. The most common way to do this is to encapsulate the mRNA into LNPs. So if we look at LNPs, they've really emerged as the biggest non-viral carrying vehicle for therapeutics, especially nucleic acids, and they're capable of encapsulating various payloads and have manufacturing advantages. If we look at the components of LNPs, it's usually composed of ionizable or cationic lipid, which is responsible for interacting with the nucleic acid cargo, which is negatively charged. The pegylated lipid, which prevents aggregation and fusion by shielding the surface of each LNP. And it's like little hands making sure that LNPs don't come too close together. Phospholipid, which is basically the main structural lipid, and cholesterol, which helps with integrity, rigidity, packing, and so forth. The main challenges in the LNP field are usually currently when it comes to immunogenicity and organ targeting, in the diversity and heterogeneity of samples, and in the instability of the lipids themselves. So if we take these components, to make the LNP particles, we would have an organic solvent that contains the dissolved lipids, as well as an aqueous solution of our nucleic acid drug substance. We would then inject these into either a microfluidic chamber, a T-joint, or something similar. And under mixing conditions, the two solutions basically interact, which allows the ionizable lipid to cover the nucleic acid and then helps form the LNP particle this way by increasing its hydrophobicity. And this is a very fast process. Basically takes a few milliseconds to mix both fluids, and playing around with the parameters, so changing the speed and ratio of the fluids injected, and can control the size of the lipid particles being produced. Now, after we make these lipid nanoparticles, one of the key things to determine the successfulness of our process step is to determine how much mRNA or nucleic acid we've actually incorporated into the LNPs. Usually, this is done by taking the LNP sample, splitting it into two identical aliquots, where one is incubated with a fluorescent dye and the other is dissolved with the detergent and then incubated with a fluorescent dye. And by comparing these two fluorescence signals, we can determine the encapsulation efficiency. Now, what we've recently developed is an analytical chromatography approach to determine one of these key LNP attributes. It's based on around the PATfix platform, except that we've upgraded it to be basically a two-dimensional analytical chromatography system where there's two different columns. One is for separation of species, specifically free nucleic acid from LNP particles, and the other one for quantifying the nucleic acid. And it has two analytical pumps as well, which allows for robust method control to both of the columns. By combining it with multiple detectors that give us interesting results, it would be the UV-Vis detector, of course, to detect nucleic acids, the MALS detector to detect the particles, and the ELS detector for lipids. And the advantages of all of this is that there's no sample preparation or handling required, as well as no dyes required to obtain quite a few results. How this looks a little bit more in detail is that we take the LNP particles, we dilute them in loading buffer, as you can see completely to the left. These are then injected on the first column, the CMAC-OH column, where the LNP particles are bound, while the mRNA it goes through the column to the second column where it is quantified, so the SDVB column. This gives us the free mRNA. Then we elute the particles from the first column. We measure their size, average size, using the MALS detector, and then we open them up to measure the encapsulated mRNA or nucleic acid species. And what we get from this is a typical chromatogram shown here, where on one end we would have free mRNA, as you can see at the top left. Then after we've done quantifying the free mRNA, you can see in the middle the LNP, black LNP peak from the MALS detector, which signals that the LNP particles are being eluted. Then after destroying them or opening them on the column, we can then quantify the encapsulated mRNA, which you can see on the right of the chromatogram. And using this basically allows us to quantify encapsulation efficiency, particle size, and so forth. But more than that, because this is a rapid analytical method, taking about 30 minutes to analyze one sample, it means that you get this critical parameter on optimizing your encapsulation at line as you're working to optimize your process, instead of sending them off to a lab to be analyzed later. This, of course, gives you a lot of freedom to quickly optimize these methods to desired results. Especially important when you're playing around, for instance, with lipid composition, the LNP solution, or changing nucleic acids and so forth. Now, to let us know a little bit more about this system, I'm going to hand over to my colleague Moitza, who will show us the PATfix LNP platform a bit more hands-on.
Welcome to the presentation of PATfix LNP Switcher, an advanced new analytical tool that enables comprehensive analysis of lipid nanoparticles. My name is Moitza Boucher, and I will guide you through its features and demonstrate its applicability with real-life examples. Lipid nanoparticles are a novel non-viral drug delivery system. Their use has enabled broader application of therapeutic nucleic acids, which are unstable and susceptible to degradation when introduced directly into the body. Following the great success of LNP-based vaccines during the COVID pandemic, LNPs are being explored for other therapeutic applications. LNPs typically consist of 4 different types of lipids and a nucleic acid payload. This payload can include mRNA, siRNA, gRNA, DNA, or some combination thereof. Due to their multiple component nature, the properties of LNPs can vary widely, making their characterization challenging. As LNP technology advances and its therapeutic use increases, higher regulatory standards are expected, requiring more detailed characterization of LNP composition and stability. Some of the therapeutically relevant LNP characteristics are encapsulation efficiency and LNP size, as well as mRNA integrity. LNPs are manufactured by mixing lipids dissolved in an organic solvent with an aqueous solution containing a nucleic acid. This process is typically carried out using microfluidics. LNPs are then diluted and buffer exchanged to physiological aqueous buffer to remove the remaining organic solvent. It is essential to thoroughly characterize the final concentrated LNP sample before proceeding with further use to ensure it has the intended properties. One of the key indicators of LNP quality is encapsulation efficiency. The most commonly used method for determining encapsulation efficiency is the modified ribogreen assay. This method is conducted on 96 well plates and is based on ribogreen reagent, a fluorescent dye that binds to RNA in solution. To determine the encapsulation efficiency, ribogreen reagent is added to 2 aliquots of the LNP sample. One of these aliquots is pretreated with a buffer containing a detergent to disrupt the LNP and release the encapsulated RNA. The fluorescence signal of the untreated aliquot is used to calculate the concentration of non-encapsulated RNA, while the signal of the detergent-treated aliquot is used to calculate the total RNA concentration. The encapsulated RNA concentration is then calculated by subtracting the non-encapsulated RNA concentration from the total RNA concentration. The encapsulation efficiency is then calculated based on these values. This protocol is very time-consuming and labor-intensive, as it requires multiple steps of reagent addition, which increases the possibility of operator error. Furthermore, the ribogreen assay does not provide additional therapeutically relevant parameters, such as mRNA integrity, and is inadequate for detailed characterization of co-encapsulated nucleic acid payload. For size determination, another therapeutically relevant parameter, a separate analysis is required using different sample preparation and instrumentation, such as DLS, NTA, or microscopic techniques. In order to speed up LNP characterization and make it as encompassing as possible, a 2D chromatography system was developed, called PATfix LNP Switcher. It has 2 analytical pumps, allowing for 2-column operation that greatly expands the separation capabilities of analytical chromatography. Samples are analyzed by an inline detector setup of UV and multi-angle light scattering detectors. MALS detector allows for detection of LNPs and estimation of their size, while UV detector enables detection and quantification of the nucleic acid payload. All this information is obtained with minimal sample preparation. The PATfix LNP switcher requires 2 columns for operation. The first stage of the process involves the use of a hydrophobic OH column, which separates non-encapsulated nucleic acid from LNPs. Subsequently, the 2 species are analyzed individually on reverse phase SWB column in ion pairing mode. This enables the separation of different nucleic acid species and the evaluation of mRNA integrity. During analysis, non-encapsulated nucleic acid passes through the OH column to the SWB column, where it is bound and eluted based on length, while LNPs remain bound to the OH column. In the next step, LNPs are eluted from the OH column, bound and opened on the SWB column. The encapsulated nucleic acid is released for analysis in the same way as the non-encapsulated nucleic acid in the previous step. When LNP samples are produced, the LNP switcher is used for characterization, eliminating the need for separate instruments that require considerable sample handling. The only sample preparation required is dilution, making the process much more robust by minimizing sample handling affecting results. This sample chromatogram shows an LNP encapsulating mRNA. The 2 signals are absorbance at 260 nanometers in red and a light scattering at 90 degrees in black. The chromatogram is divided into 4 sections, corresponding to the 4 steps of the analysis: load, non-encapsulated mRNA analysis, LNP elution, and encapsulated mRNA analysis. In the light scattering signal, a peak is observed in the LNP elution section when LNPs are eluted from the OH column. This signal allows for the estimation of LNP size. In this demonstration, we will focus on the information provided by the absorbance signal. A peak is observed in the first analysis, representing non-encapsulated mRNA, while the absorbance peak in the second analysis corresponds to encapsulated mRNA. The concentration of both mRNA species can be determined from the calibration curve of the mRNA standard, based on the absorbance peak area obtained by integrating each peak. By comparing the concentration of both peaks, encapsulation efficiency can be calculated. This method of quantification requires far less sample handling for preparation than the ribogreen assay. When LNPs are used to co-encapsulate two different nucleic acids, the ribogreen assay may not be sufficient for characterization purposes, as the encapsulation efficiency of the two nucleic acids may vary. An LNP co-encapsulating mRNA and pDNA in a 1:1 mass ratio was prepared. When analyzed on the PATfix LNP switcher, the following chromatogram was obtained. The light scattering signal is comparable to the previous example, enabling the estimation of LNP size. However, the absorbance signal displays additional peaks. Due to the presence of two nucleic acids in the sample, namely mRNA and pDNA, two distinct peaks are observed in both the non-encapsulated and encapsulated analyses. This way, encapsulation efficiencies for both nucleic acids can be determined independently. From areas of the first eluted peak in both analyses, representing mRNA, its encapsulation efficiency can be determined. Similarly, areas of the second eluted peak can be used to determine pDNA encapsulation efficiency. This analysis provides crucial information for LNP development, as produced LNPs do not contain mRNA and pDNA in their original ratios, which could impact the therapeutic effect of the LNP. Such detailed data cannot be obtained with any other assay known to us. The PATfix LNP switcher represents the next key upgrade in the analytics of nucleic acids, enabling the analysis of more complex samples, such as LNPs, for which the traditional approaches are often lacking. By minimizing the required sample preparation, the PATfix LNP switcher offers a novel hands-free approach for LNP characterization. In addition to determining sample concentration and encapsulation efficiency, it provides valuable insights into mRNA integrity and LNP size estimation, all within a single assay. Furthermore, it can be used for analytics of co-encapsulated payloads. To learn more about the PATfix LNP switcher and its capabilities for LNP characterization, visit our website, try out the software demo, and contact our team for more information.
Thank you for a fantastic presentation. Next up, I would like to pass over to Dan and to Tristan.
So I'm going to talk today about next-generation lipid nanoparticles for RNA vaccine and therapeutics. And the future is already here. As you can see, 23 years ago, Mr. Bean here already understood what's the difference between RNA and DNA, and he needs a good template, somebody to copy from. And the lady, the mother of dragons, already showed us what's the difference between double-strand DNA to a single-strand RNA here 10 years ago. So the central dogma of biology, basically going from DNA, to RNA and proteins, have been around for many years. And it's now the next generation that we target the RNA, and we want to target it specifically to specific organs, and within the organ, specific cell types. So we are living in the RNA arena now, that we can do many things. We can silence genes, we can activate them or express them, we can edit them, and in a very near future, we will be able to replace them. And what can we do with this? So we can silence genes predominantly with SI, small interference RNA. We can activate them with a messenger RNA, and we can edit them and replace them by many types of genome editing tools. For example, the CRISPR-Cas9 one that reached the clinic, and we have already a very nice clinical example, which is Cascavy, that is clinically valid as a new drug therapy modality. So the challenges in RNA therapeutics is twofold. There is the RNA itself, and I'm sure you already heard a lot about RNA impurity, but it's also the delivery issue. So we need an appropriate delivery system. And the requirements are as follows. We need an efficient RNA encapsulation. We need evading clearance mechanism, avoiding toxicity and immune activation by the carrier with the RNA payload. The carrier must internalize, and the RNA must be released. And then, of course, we need specificity. So what we're going to talk today is about lipid nanoparticles that being a major player in this. And we already have 4 clinically approved drugs and vaccines. The first one on Patro, an alnylam drug, was approved with lipid nanoparticles and siRNAs already in August 2018. And the siRNA is encoded here basically against the TTR amyloidosis. And then we have 3 vaccines, the 2 COVID vaccines by BioNTech, Pfizer, and by Moderna, and an RSV vaccine by Moderna. We have 4 lipids, sometimes more than 4, in a formulation with a favorite RNA, and it has a lot of advantages. It's protecting the RNA. The system is completely biocompatible, biodegradable, protecting the nucleic acid, increasing the stability in circulation and therapeutic index. And of course, as I mentioned, some of them are clinically approved. In my lab, we are interested in combining first RNA delivery system, including also protein engineering, with new potential drug targets. So we know how to do many things. We know how to silence genes with RNA. We know how to activate them. We know how to edit our genome with RNA, and hopefully in a very near future, how to replace genes. As a vision, we are living in the revolution. And here we're going to hear a lot of examples today about purity, about the importance of being able to control your analytics, which is very, very important. But it's not only the delivery, as I mentioned, it's also the payload. As an example for that, we are collaborating in being able to open, close genes, or to be able to control their translation, and of course, their stability. In this sense, the integrity of the payload, the integrity of the coating with the lipids is super important. Another example that we recently published is the ability to use circular RNA as a new modality inside LNPs. But still, the open questions are endosomal escape, which is a bottleneck for LNP-mediated therapeutics, and understanding the intracellular immune response of lipid nanoparticles. All in all, we're going to hear today lots of talks about the importance of analytics, how we can control each step, and hopefully that will give us the next generation LNPs. I want to thank you for listening, and I have enjoying your time.
Hi, everyone. So my name is Tristan, and I work in Sartorius BIA Separations, and I would like to sincerely thank everyone for bringing this together, and especially now to Dan for showing us and talking to us about really what are LNPs, what's their current status, and what's their future. And really, there is many things you can do with LNPs, but you at some point you want to bring them towards commercialization, and then where you've done all the great work in biology and all of that, and then you want to really be sure that what you are getting is robust and reliable. So that's where you have to think about more intensely about the characterization and the other manufacturing challenges that you are facing with the LNPs. And when you do that, you face certain challenges, and people have faced them, and some vaccines and therapeutics that are already on the market, like Dan described, have at least partially solved those problems. But let's talk about what they are. So there is the inherent kind of instability of the particles, because LNPs are a supramolecular assembly that kind of all the weak forces, they make together the particle, they hold together the lipid envelope, and they hold together the interactions between the RNA and the lipids. But those are weak forces that are susceptible to their chemical and physical environment. And then that brings about the heterogeneity, which is a consequence of that, but is also a consequence of other factors on the molecular level. And then it's also a consequence of the stability and the storage of the samples. And then because these LNPs are very modular, so you can change out the ionizable lipid, you can change the lipid composition, and so on. That brings about diversity, that it's then, you can bring about subspecies of different LNPs, to say so. And you have to think about that when you're developing, when you're thinking about, let's say, the stability and the characterization of your LNP. And then because in a simple system of LNPs, let's say you have four lipid, so 4 different lipids, like Blaž described at the start, and then you have the payload, like one RNA. So you have 5 different molecules that are interacting with each other, and that brings about also their interactions. And on the molecular level, that can bring about degradation of each molecule, and then interactions of those further on. And all of that, even inherently, but also because of those stability issues, can give you complex morphology and not just very simple spherical particles that we maybe like to think about. Now, in characterization, then there's these challenges. Because of all those reasons, you have challenges that the analytical methods currently available are poor predictors of in vivo and in vitro behavior. And that generally means that what we are looking at is not really the most important part, or at least it's not the whole part. So that what we get as our analytical results, characterization results, is not the whole picture. Or we are changing the system too much by wanting to look at the particles. So that's why I'm saying here that for intact particles, there are very limited reasons on how to analyze them without any pretreatment or any labeling, because labeling is a very powerful tool on each level, on like when you have the particles formed before, or just for labeling RNA. So many different ways to label your species, especially with fluorescence, because it's very specific and it's very powerful. But we have to realize that all of that changes the system a little bit. Let's say if you're using fluorescent RNA, that might be physicochemically very, very different to what you might have if you didn't have that. And that's that's why we want to kind of strive towards non-labeling techniques, like maybe the more developed pharmaceuticals are using. And that's why, because of all those reasons, we then have a kind of a complicated CMC, so chemical manufacturing controls for our drug product, and we want to strive towards solving that, and having things that are applicable and that give us the results that can predict how our product is going to be acting. So the analytical platform that we have developed, that has been described quite a lot in this video already, so I'm going to go through this quite quickly. But it's like a 2-dimensional chromatography setup, and in the first part of this, the OH column is a hydrophobic interaction chromatography. So here we are utilizing the hydrophobic properties of particles such as LNPs. Basically, the larger the particles are, the more they stick to the column, because they have a larger hydrophobic surface. And even then, if you compare them to non-spherical particles like RNA, that is also polyanionic, it doesn't bind to the column as well, so it gets separated from the LNPs based on their chemical properties. And then you detect both of those species with the appropriate detectors. So for the particles, for the LNPs, you would use multi-angle light scattering. So you just direct light scattering information by observing your LNPs. And for the RNA is the UV-Vis detector that detects the chromophores in your RNA. So the nucleobases are very UV active, and it's easy to detect RNA this way, but you do have to separate it from other things beforehand. And that's where the SDVB column comes in. So we separate it from particles in the first step, and then in the second step is reverse phase. Ion pair chromatography is the mode used in this step. And here is where you're utilizing the you're ion pairing the phosphate backbone of RNA, and then you're utilizing the hydrophobic properties of that. Here also, the larger the RNA is, the more of those ion pairing reagent it's going to have on there, the stronger it's going to bind. So you're separating by size. But then additionally, not just that, you're also separating by hydrophobicity. And then you can separate away some other species that might be important in characterization. And just a quick setup of how this works. So you bind on the OH column the LNPs, you analyze them on the MALS detector, and then RNA, you use free RNA, it goes past here and it's analyzed on the SDVB detector, while the encapsulated RNA and then binds to the SDVB column. That's quite important, because this is kind of an online particle opening that we have developed. So when LNPs get eluted from the OH column, they bind to the SDVB column very strongly. And we figured out that you cannot elute them as intact particles because the interactions are too strong. So what you do is that you, with a high temperature of 60 degrees and an organic solvent, you basically dissolve the particles into its separate components. So this is how we can do with no sample pretreatment, because we are opening the particles on this column with the interactions with the solid state of the column and the percentage of organic solvent that we then use to do those separately. And then you quantify the encapsulated RNA by that disruption of the particles, and then you also can quantify the lipids if you put in another detector like the ELSD or the charged aerosol detector behind that. So the first parameter you can determine is encapsulation efficiency by comparing those. That has been described. And then a very important thing is that if you have co-encapsulated species, you are going to be able to separate them, which is quite impossible with like an assay-based determination of encapsulation efficiency. And this is not just important for encapsulation efficiency of those, but also for their integrity and for any other impurities that you might be forming. And for this, it's basically necessary to have LC-based analytics, chromatographic ones, so that you are able to separate those and quantify them. And then very important in applications for gene editing, like Dan described before me, for kind of CRISPR applications, where you need to separate away at least your Cas9 mRNA and your small guide RNA. And that is possible to do on a setup and with a chromatographic approach. Then in terms of multi-angle light scattering and the kind of particle size and its integrity and so on, we have different methods that can be applied. So here we have, for example, NTA, DLS, cryo-TEM. Some of them, like cryo-TEM, are very powerful, but they are very laborious and kind of hard to implement as a critical quality attribute, also not very quantifiable. Now, DLS is the most widespread one. It's quite good for, you get very fast results, but you can get very misleading results, because a very large particle will give you a much larger response, and that can kind of skew your results towards larger average values because you are not separating subspecies. So that's why it's also good to do subspecies separation with certain techniques one might use. In this case, we can present the separation using hydrophobic interaction chromatography. So the larger particles bind more, and you get a sort of a separation. And you see here, if we see a plot of radius, we can see an increase by size as this peak gets further on. So we are separating these particles by size, and we can show that it's comparable to microscopic techniques like NTA, but importantly is that we also, by that, we remove away the larger particles, and then we can see subpopulations of particles. And here is an example of that. This could be obviously further, whatever you need, whatever you're expecting from your particles, you will want to change maybe those different ones, you change your method a little bit so that you want to see some subpopulations that you're expecting or you think are problematic. Now, another very important thing, critical quality attribute of the drug product is mRNA integrity, because if your RNA is not intact, you're not going to be able to get it into the correct protein that it needs to be translated into in your body. And with the larger your cargo gets, generally the more unstable it is. If we're talking about mRNA, and then, for example, saRNA, self-amplifying, is even larger, even more unstable. And then the smaller ones like siRNA are more stable in terms of that. But let's say if we focus on mRNA for now, it's quite unstable. So part of the reason, a major part of the reason why we are using drug delivery systems like LNPs is that we want to protect it from that kind of instability. Maybe reduce the water around it, so reduce the hydrolysis a little bit, but then in the body also protect it from the RNases that will degrade it. And here, the important thing here we have on the LNP switcher is that we can separate away. This is one crucial thing that maybe if you're using capillary electrophoresis needs to be looked at. So if you're opening the particles and you have a lot of free RNA or just even some free RNA, that one is going to be more unstable. So it will be more degraded than your encapsulated RNA. So you might get a value that's it's not real, and that will give you might then accumulate some different percentages of how intact your RNA is, and it doesn't correlate with your biological activity. Might be because of that also. So if you're separating the free from the encapsulated beforehand on some sort of a chromatographic system, let's say, and then you're using that to analyze the integrity, that would be much more precise and accurate in terms of doing that. So on a switcher, because we're separating the free and encapsulated RNA, this is inherent to the system. And then we have there's an article how to do this using reverse phase ion pair chromatography to determine the integrity of your payload of each separate one. So separately for free and separately for the encapsulated. And you see for free RNA, this is like at high temperature, 35 degrees, you're increasing, it's very fragmenting very quickly, while for the encapsulated RNA, it's fragmented quite slowly. And you can see that it's more protected than the free RNA. Now, another crucial thing that's been going on in this field is the mRNA lipid adducts. Now, this was, I'm showing here from an article by Hashiba in this year, is that aldehyde impurities that are forming from your ionizable lipids, they can form as oxidative hydrolytic impurities during stability, or they can also form, they can also bring certain or similar impurities like that from your synthetic route of your ionizable lipid. So you get you would get degradation, and then you would form aldehydes that can alkylate your RNA and can render it inactive, or can even maybe form certain side products and so on. So you can use, so it is quite important to use reverse phase chromatography to analyze species like this, because capillary electrophoresis is not able to quantify this because they are, by size, they're quite similar to the RNA, but by hydrophobicity, they can be very different because those long lipid tails attach covalently to your RNA. And here you see from the first article on this from Packer in 2021 at Moderna, you can see that the correlation between CE integrity and protein expression is not very good, while the correlation between reverse phase ion pair analytics and relative protein expression is very good, actually. So it's the integrity based on LC in this case correlates much better with the actual activity. And here we can see, this is an example from our analytics. We have a capillary electrophoresis on the top and SDVB, so reverse phase ion pair analytics on the bottom from the LNP switcher. And we can see that by CE, the integrity of this RNA is 74%, whilst by SDVB is only 47%. But if we combine the intact part with this part, the adduct part, we get the same percentages here. So basically, those two are co-eluting in the CE because they are similar by size but very different by hydrophobicity. So that's why it's important to monitor those by reverse phase ion pair chromatography. And this is very important for stability studies because generally the trend would be to have some sort of a fridge stable LNPs. But if you are then here, you see we are increasing by temperature, we're increasing this percentage of adducts. So that is something when you're developing those conditions for stability, you will definitely want to look at, because at -80, those are much smaller than in a refrigerated temperature. So the LNP characterization is a growing field that demands new analytical methods that could be used to write reliably and widespreadly. Some of the ones that are used right now are maybe limited in what they can give you, and maybe can give you some false positives or false negatives. So that's why you want to spread out and analyze your product further, but you want to use some certainly more advanced techniques like I described before, cryo-TEM or maybe SEX or things like that when you're developing, to really see how your product looks like. But then for further on, you will want to use methods that can be used on basically daily basis for analysis when you're moving to a large scale commercialization of products. And that's why also here we're introducing and developing LC-based methods that have a long history of doing that and are able to, especially are powerful for separating subspecies. And this is what this sort of a system can do for you. It can analyze a drug product, an RNA LNP drug product, from many different aspects. So I would like to thank everyone from our company for doing this work and for helping us with this work, especially our department, the PC4 Process Development Analytics Department, that some of them you have seen already in this session, and I really they are all responsible for this work. So really, all the thanks from my side to them. And then also thank you again to Dan Peer who is a pioneer in this field, for presenting and showing us what holds the future for this field and how powerful these modalities can be. Thank you.
Well, thank you everyone for some great presentations. Before we start our Q&A, we would just like to ask the audience a quick question, which you should see pop up on your screen now. Great, thank you for your input. And now we're going to start our Q&A. Please keep your questions coming in, and we'll get to as many as we can, and we can answer any others by email. So first up here, I have a question. Which CQAs are most overlooked right now in RNA LNPs? And what is your advice on implementing the appropriate analytics? And Blaž, if we come to you here first.
Oh, thank you. Yeah, I think given where the field is moving, especially with the implementation of CRISPR-Cas into the LNP platform, distinguishing between multiple co-encapsulated nucleic acid is something that is going to be explored more and more, because of course the underlying activity of the LNP particles is going to heavily depend on the ratio between, for instance, the guide RNA versus the Cas9 mRNA.
Yeah. Okay.
Tristan, what would you say?
Yeah. Thanks, Bash. Yeah, you're right about that for sure. So that means that we need to currently in the kind of USP guidelines for critical quality attributes is obviously the encapsulation efficiency and the quantification of RNA, and that is currently based on a fluorescent based assay like ribogreen. But as described, as Blaž said, especially for more complex multimodal cargoes, you need to use something that can separate species. So you will want to use something liquid chromatography based in order to be able to separate and quantify those. And additionally, as I described before, the product related impurities. So the impurities that form after the drug product is formulated, so something that you cannot detect before by analyzing the drug substance or the excipient. You will also need to use techniques that can either do this on an intact particle or disrupt the particle and analyze it. So an example of that is the lipid RNA adduct. This is also one crucial thing that needs to be added. So that you can fully comprehensively characterize your sample.
Fantastic. Thank you both. Another question here. What are the limitations of current analytical workflows in characterizing stability and predicting activity? What could be the consequences of that? And what would be your advice on how to tackle these issues? And Tristan, if we come to you here first.
Yeah, thanks. Thank you for that question. So, as I said, most of these analytics right now are assay based. So those are kind of maybe the simpler ones that can be implemented, and also they have a history of showing good results. But they can have problems with robustness and reliability of samples, and especially when you come to more advanced samples that needs some sort of a separation. So, and because this is a young field, maybe we need to take some notes from the more developed pharmaceutical fields and maybe go towards the implementation of analytical techniques that have shown to be good in high throughput, that are precise, and that are accurate when developed properly, and really look at those and then move to thinking with commercialization of your product in mind to implement those.
Fantastic. Thank you, Tristan. And Blaž, would you like to add anything on this one?
Yeah, I would say a thing that's often overlooked here is when it comes to cargo integrity, current methods rely heavily on sample handling that can lead to errors. So, as Tristan said, basically implementing methods that require less sample handling, which can introduce these errors, is, I would say, of great value to the field.
Absolutely. Well, thank you both. And another question here for Tristan. Can you detect degradation products in LNP formulations?
Okay, thank you for the question. Yes, one of those, if we're talking about, it's a few things you can talk about. So one of them I already described, which is the RNA lipid adduct. It kind of stems from the lipid degradation. So the ionizable lipid degrades and it reacts with the RNA. So that's one part of the degradation product that can occur. The other degradation products are the shorter fragments of RNA that are formed when RNA is degraded. So that also can be done. And then the third thing is that other degradation products from lipids that do not react with RNA, then that can also be done. But then for lipids, we generally need another detector, which would be a charged aerosol or a ELSD detector, so that we can observe those small molecule degradation products as well.
Great answer, thank you. And Tristan, another question here for you. What size nucleic acids can be analyzed using this setup?
Okay, thank you. So the setup, kind of in a basic setup, is mostly used for mRNA, but that can easily be done for, let's say, RNAs down to 50 or 50 nucleotides, up to even the size of siRNA, up to, let's say, 10,000 nucleotides. But even if the RNA is larger or smaller than that, the ion pairing reagents and certain conditions can be changed to achieve binding of all sizes.
Fantastic, thank you. And Tristan, another one here for you. What different lipid compositions have you tried analyzing with the LNP switcher?
Yeah, so we have tried many different ones, all the ones that are the most common. So from the current vaccines that exist, those compositions, then certain ones that you can buy off shelf online, like the pre-mixed lipid mixtures, and then also certain other types of additional ionizable lipids and cationic lipids that can be purchased. We have also done this with other nanoparticles like liposomes.
Fantastic, thank you. And I've had another question come in here. Thanks for a wonderful presentation. I would like to know if we are able to quantify free lipids after breaking down encapsulated mRNA in PAT system. I didn't see lipids peak after breaking down encapsulated mRNA to quantify mRNA.
Yeah. That's a great question. So that's definitely possible to do. We have another publication regarding this that can be found on our website regarding lipid composition and RNA. But yeah, you need to add a third detector, a charged aerosol detector or evaporative light scattering detector. And from that one, then, because with chromatography, you're able to separate away the RNA from the lipids, so that then you're able to quantify first the RNA with the UV detector and then the free lipids with the other detector, and separately and quantified.
Fantastic, thank you. Well, thank you for answering those questions. I'm afraid that's all we've got time for today, but any questions we didn't get to, we will reply by email. This webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the media tab on the left-hand side of the screen or in the on-demand email. So all that's left is to thank Dan, Tristan and Blaj once more for some great presentations, and thank you to the audience for watching. We hope you'll join us in our next session, which will be covering accelerating AAV process development. This will be starting shortly, and you can find it by returning to the event lobby. Thank you.
Thank you.